Alex Popham, the Breckinridge County 4-H agent. I'm over here at the Extension office again today. We are part of the University of Kentucky's College of Ag, Food and Environment's Cooperative Extension Service. And this particular video is a continuation of our 4-H set activities, so just exploring science, engineering, and technology together. Today we're going to be focusing on microscopy, so we're going to be learning about microscopes and then getting to try out a few different techniques for making slides. So we're going to start out with our first piece of equipment here. This is a pretty basic microscope. It's referred to as a stereo microscope, and you could magnify pretty much any object you can hold in your hand. So our 4-H members tend to use this scope in Bug Club to look at insects. I'm sure our Outdoor Explorer Club will get to look at some minerals and rocks up close, as well as leaves and other things in the environment. So it's a really cool microscope, but if you want to look at things up really close and thin layers, you're going to have to check out our compound microscopes. Now, these particular pieces of equipment are called compound microscopes, and they do require slides. That's how you're going to look at your specimens. They have to be mounted and placed on a slide here on the microscope to check it out and magnify it. So I'm going to use this third microscope, just a larger compound microscope, as an example and walk you through the different parts for those that might not be as familiar or used a microscope recently. So these two pieces, I'm going to start from the top and work my way down. These two pieces are the eye pieces. And you can see on the side, they're marked what their magnification is. So this eyepiece, all by itself, will magnify an object 10 times. And that's going to be important in a minute when we look at the total magnification of an object. From the eyepieces, moving down the body tube, you'll run into the rotating nose piece. And on that nose piece are different objectives, different lenses of different strengths, and they also mark their magnification. So the strongest magnification here is this oil immersion lens that will magnify an object 100 times. So when you look through the eyepiece, you have to take the magnification of your objective and times it by the magnification of the lens. So in this eye here on the right side, since it's magnifying 10 times, you're going to take 10 times 100, and that means that object is going to be magnified 1,000 times. So you're going to get to see it up pretty close. Now the specimen on the slide will sit here on the stage, so this piece of the microscope here. And if you look down into the stage, you'll see the diaphragm that the light passes through, and the source of the light is down here on the base. So the light source is sitting on top of this microscope base that has the power source. And then the last thing I'll point out is how to actually get your object, your specimen in focus, and that's using these two knobs here on the side. So we have a coarse adjustment, which moves the stage up and down a lot farther than this fine adjustment. And you'll use both to get an object or your specimen crystal clear and in focus so that you can examine it. So now that we've reviewed the parts of the microscope, we're going to try out a few different techniques for slides. And a quick shout out, a thank you to Miss Bev here at the office for letting me steal some of her flowers. We're going to start out by actually making a flower petal slide. The first thing you'll need is an actual microscope slide, a piece of glass, and a cover slip. And they need to be clean. You don't want to get your fingerprints on them. You don't want dust or debris on them. So wiping them down with an alcohol wipe works great before you get to using them. So I'm going to start off, I'm going to do a wet mount, and I'm just going to put a drop of water in the very center of our microscope slide. And then I'm going to use an X-Acto knife to cut off a piece of the petal. And I'm going to use some tweezers to just very carefully set it in that water. Oh, it wants to stick to the tweezers. There we go. Now the hardest part of making any slide I've found is getting the cover slip on there without adding adding air or air bubbles to your specimen, which makes it really hard to see when you get it on the microscope. So we're gonna try to be as careful as we can, and I'm always learning and it takes practice. So we're gonna put that cover slip at a 45-degree angle, and then we're just gonna very slowly, using a toothpick to help us keep it going slow, we're gonna drop it down into our specimen. Okay, and you can press it very gently. You don't want to break the slide cover. Okay, and I will post pictures to see how our slides turned out. Did I get a bunch of air bubbles in it or not? So I'll share pictures on the next post, and we'll be able to check out our final results. So moving on, instead of doing a mount with water, we're actually going to dye a piece of onion skin, the epidermis. For this slide, I'm going to need to put some gloves on just because I don't want to stain my hands. And we're going to be using a really common dye in biology labs. It's methylene blue, and it only takes a drop or so. So to start off with, I need to just break off a small piece of onion, and I want the inner layer. It will end up with a thin, paper-thin piece of that layer, and that's what I'm aiming for. So here I have a pretty small piece I've just broken. You could cut it with a knife, but I just find tearing it with your hand works great. And you can see that there's this thin layer laying on that inner top surface, and I'm just going to use my tweezers to pull it off. And I'm going to try to not let it fold over on itself because I want the layer to stay nice and thin. And I'm going to put it in the very center as best as I can of my slide. And you can always use a toothpick to help, place and keep it from folding over on itself. Now I am going to add a drop or two of the dye. You could add water, do a wet mount, and then place your cover slip on and just drop some methylene blue on the side of the cover slip and let it flow through. But I'm just going to stain it directly today. So I'm going to add a couple drops. Now this particular dye, the methylene blue, is a positive dye, so it's going to be attracted to all the negative parts of our onion cells. So hopefully we'll get a good view of the membranes and then the nucleus. That's the plan. All right, next I'm going to grab my cover slip and we're going to try to set it on here, again at a 45-degree angle, and drop it as slow as we can. Now you can see that it's swimming in that dye, and I want to clean up my specimen. I'm going to let it stain for a little bit while I'm talking, and then I'm going to use a paper towel and put it along the edges to help soak up all that excess dye. So I'm just tearing right now a piece of paper towel, and I'm going to set it on the edge and let it soak up all that excess. And this will make it a lot easier to see. It will make your slide nice and neat. And you'll just continue until it's dyed, but it's a nice clean slide mount. So I'll continue to do that after the video and get that all nice and clean and ready to check out under the microscope. All right, our last slide, I'm going to need to change gloves. And I am going to mount some of my own cheek cells. So I need a clean toothpick. And of course there's the sharp end and the blunted flat end. I want to use the blunt end in my mouth. So I'm going to hold the sharp end and I'm just going to rub it on the inside of my mouth, up and down like 12 times is fine. So hopefully I've caught lots of cheek cells on the end of that toothpick. And I'm just gonna place a drop of that dye in the center of my slide. And then just mix my cheek cells into the dye. Like so. And then again, the last thing is to put my cover slip on. So I'm using a new toothpick to just, at a 45-degree angle, drop that down as slow as possible. Oop, caught on my glove. All right. And again, I'll clean it up. I'll stick it under the microscope and post the pictures sometime after I post this video. That way we can see the results from our microscopy experiments today. So definitely check this out. These are easy to do at home. You'll just need some simple supplies or definitely contact your extension office and see if you can check out the resources or the curriculum that goes along with this. So thanks so much for joining us today, and I'll see you next time.